AmeriFlux observations to improve E3SM land model predictions using machine learning. E3SM stands for the Energy Exascale Earth System Model, which is the Department of Energy's Earth System Model. And you'll hear me refer to ELM, which is the land surface component of that model. So a little bit in terms of our motivation here. So ELM is, as some of you may know, an increasingly complex model. So it's a full land surface model. It does the energy balance. It simulates hydrology with 10 soil layers, biogeochemistry with the cycling of carbon, nitrogen, and phosphorus, and also things like land use change, urban systems, agricultural systems. And so in order to quantify uncertainties or do calibration in the model, typically we need to run large ensembles, many simulations, for example, with varying parameters. But the model is generally pretty slow to run, even at a single grid cell or point, and it requires, if we're doing the biogeochemistry part of the model, long spin-up. So we equilibrate the carbon and nutrient pools using pre-industrial conditions and then run generally starting in 1850 up to present day with varying CO2 and other factors. And so what I'll talk about today is how we're using an approach called surrogate modeling to increase the efficiency of uncertainty analysis of things like sensitivity analysis and calibration that we want to do. Okay, so just a little diagram of how we're doing that. And so typically we have some uncertain input parameters. I'll talk a little bit more about some of these in a specific example. And we take those, we vary those, and we run ensembles of ELM, and that produces lots and lots of output. So the model by default, I think, produces something like 400 output variables, and those vary if we're running regionally or globally over space, and of course, over time as well. And so we use those ensembles then to generate these surrogate models. And that allows us to do uncertainty propagation. And so what's a surrogate model? And so for the purposes of generating these models, we treat ELM as kind of a black box where we have a number of input parameters that can vary. We run an ensemble of generally a couple hundred to a couple of thousand ensemble members. And then after that, we never really need to go back and analyze that ensemble directly. Instead, we construct a surrogate model for specific outputs of interest. So for example, if we're interested in gross primary productivity, we take all of those outputs over that parameter space and we construct basically fit a model to the model output. And the reason why we do that is once we have that, it's generally several orders of magnitude faster to evaluate that surrogate model than it is to run the original ELM. And we need much denser sampling of that model for things like sensitivity analysis or model calibration. For example, Markov chain Monte Carlo, which is a common method that's used. So we have the problem again of so many outputs, even considering one output like GPP. If we're looking at a time series of that, we would have to generally go back and construct a surrogate model for each one of those time points and spatial points. And so we're using a technique called Carhoonen-Loewe expansions to help reduce the dimensionality. And the reason we can do that is there's very strong, as you know, spatio-temporal correlations in these model outputs. So for example, GPP has a lot of spatial and temporal autocorrelation. So this is similar to what we might do in like a principal component analysis. And so the surrogate model in this case is an artificial neural network that takes as input the same parameters as ELM. So for example, parameters that control photosynthesis. Instead of typically an output would be, for example, GPP, but in this case, we're taking that and we're doing this dimensionality reduction step. And so we're building surrogate models of these eigenfeatures. And given those strong correlations in the data, we're able to capture most of the variance in those outputs with just a few terms, say 10, for example, and that allows us to reduce what we would have depending on the spatial and temporal resolution of the outputs, going down from thousands of outputs to 10 or so. And so here's a specific example where we ran an ensemble, a global ensemble of ELM simulations focusing here on gross primary productivity. And we varied 10 parameters using the 275 ensemble members. This is actually a pretty small subset of overall. The ELM parameters has more than 150. So these are selected due to their expected influence on GPP. Temperature sensitivity parameters, and a few others. So we've defined minimum and maximum bounds for each parameter based on literature, or in some cases, just expert knowledge or guessing. And we randomly sampled over all of those supplied ranges. And so what we can do then, once we have a surrogate model that's constructed based on that ensemble, we can apply sensitivity analysis. And so what we're looking at here on the top is from the model simulations selected at the Harvard Forest grid cell, and how the sensitivity of those 10 parameters varies over time. And so what we see, this blue, so the height of the bar is kind of the relative sensitivity of that particular parameter and its control or, on the variations of GPP. And so this FLNR parameter tends to be pretty dominant. At all times, at least 40% of the sensitivity there. But we can see a seasonal cycle there of how some of the other parameters become also influential. This MBB opt, which is the ball-berry stomatal slope conductance parameter, becomes more important during the growing season, tends to show up more when things are starting to dry out a little bit, for example. So we can contrast that with the California site here, the Sky Oak site, where this red and the green, so these stomatal slope and intercept parameters tend to dominate the sensitivities at this much drier site. And just a couple of more, so sensitivities of tropical sites, and we can see in Brazil, this is a Santarém site, a rainforest, where it's maybe a little bit less of a seasonal cycle, but still kind of a wet, dry dependence, where this MBB opt or stomatal slope parameter is sometimes more important and sometimes less important. And in a tropical savanna here, where we see this yellow one, or the rooting depth distribution parameter, becoming quite a bit more important. And so we can see these sensitivities vary over both time and space quite significantly. And since we performed a global ensemble and we have a surrogate model that was constructed based on those global outputs, we can start to look at these sensitivities over space. And so I've selected just one parameter here. This is the stomatal slope parameter, and we can see the movie works, how those vary over the course of the year, the seasonal cycle. And so we can see, for example, some areas like the Sahel, that parameter is always quite important, but is varying a little bit in significance. Whereas in other areas, there's a pretty strong seasonal dependence of the sensitivity of this parameter. And so why are we doing these types of analyses? I think it's important in a couple of ways. And so a model like ELM, as I mentioned, has a lot of parameters. And so this can help us reduce the number of parameters we need to optimize, because the optimization problem becomes basically exponentially more complex or expensive as we add parameters. And also, if we know a particular parameter is sensitive at a certain time or place, that can guide observations as well. So those parameters are associated with specific processes. And we don't need to see that again. All right, and so now we can start to think about model calibration. And so we have our ELM simulations that we've trained this, what we're calling KLNN, or the Carhune-Love neural network surrogate that's been trained on those outputs, and applying Markov chain Monte Carlo to do posterior sampling on that using the Bayesian likelihood function. And we can use the flux observations as constraints. So we're trying to minimize the difference between the models and the observations. And so here's an example of at Harvard Forest, what we see is the model prior here, represented by this kind of red envelope. So that's basically selected at this grid cell, the variation or the uncertainty that we have in GPP when we vary the parameters across that those entire ranges that I showed earlier. And the green is the model posterior, so trained on, calibrated against the FluxNet data observations. And so the envelope of the green here is much smaller, indicating that our posterior probability density function of that output has been as much smaller or much more tightly constrained. And won't go through all of the specific parameters here, but this method gives you those density functions for each parameter as well. And so we can see, if we zoom in on that, the FLNR parameter, or the fraction of leaf nitrogen from visco, is quite tightly constrained, which makes sense because that's one of the most sensitive parameters for GPP. So we have observations of GPP, that's going to constrain that parameter generally more than the others. And this model slope term, MBB opt, is also constrained as well. But if we look at some of the other parameters, they're really, those distributions are nearly unchanged from the prior, which means there's really not a lot of information in that observation, in these observations about those. And we would need to bring in other sources of information to constrain them. And so we did that for 96 AmeriFlux and FluxNet sites. And so what we're looking at here is sort of the purple line showing relative errors in terms of the using the default parameters in ELM. The red showing the mean. So over that ensemble of 275 simulations, looking at the difference between the model and observations, that ensemble mean. And then the green line is after the calibration from the posterior estimates. And so we can see at all of these 96 sites, we've significantly reduced those relative errors through calibration. And then on the bottom here is the sort of the prior and posterior ranges of this FLNR parameter. So the prior is given by this big black bar. So that's the entire range that we sampled over. And then for all of the 96 sites, these sort of posterior calibrated ranges. And so kind of, you can't read the individual site names, but we've tried to group them here by the ELM plant functional types. And so the first, these blue ones here represent evergreen forests. The green represents the tropical evergreen sites. I think deciduous systems are over here. And so we do see, and what we do when we run ELM is we sort of typical global simulations, we assume a plant functional type has the same parameters everywhere. And so when we do this calibration, we see that's not necessarily true, right? So even with these sites of the same color, there's a lot of variation in this sort of calibrated value of FLNR. So I think that needs more investigation in terms of whether or not how well we trust these results, and whether or not maybe we need to consider, a more fine grouping and more plant functional types to account for some of this variability. We can also begin to bring in other aspects of uncertainty, and so a big one is model structural error. And so the results I showed just before don't include this. My colleague Kachik Sargsyan is pioneering this approach where we can bring in estimation for a model structural error by not requiring very many changes to the code, but instead altering the likelihood function in the calibration. And so this allows for partitioning of uncertainty. And so we can see this example at the Park Falls site where we have a calibrated posterior best simulation, but we have this kind of light blue that represents the uncertainty in that result that stems from model error. And that's bigger than other terms. So the darker blue is the posterior uncertainty from the remaining sort of parameter uncertainty after calibration. And the surrogate error, so the error that results from the surrogate not being a completely accurate representation of ELM, is hard to see there. So in this case, the surrogate model is quite accurate. And so the model error tends to be the dominant uncertainty, kind of pointing to places where we might need to improve ELM. And hopefully this can give us information in the future about which models or submodels or parameters are responsible and where we might target further model development. So in summary, I think surrogate models allow for efficient sensitivity analysis and calibration with a limited number of ELM ensemble members. So this really helps in terms of computational efficiency. And this approach where we can train the neural network surrogate in a reduced eigenspace allows us to really get at these spatiotemporally varying outputs, which has been a real challenge in the past. We're currently applying these techniques for other versions of ELM. So this was a very focused example just on the photosynthetic parameters. There are crop versions of ELM, there's SPATE, which brings in vegetation demography. There's a methane submodel. So we're interested in calibrating those parameters associated with those processes as well. And then just a little bit there about some of the resources that we're using. So we have a tool called the offline model testbed, which allows running single grid cell ensembles and uncertainty quantification calibration. Kind of if you're interested in running at a single Flux site, it's relatively efficient to do that. And then there was a workshop last fall hosted by the NG Arctic project where they spent a long time developing a Docker enabled version of ELM, which allows you to get the model onto your own laptop and begin to experiment with the model and test that. So thanks, and please let me know if you have any questions. Yeah, so we have time for.
And then thank you for the talk. This is super interesting, a very clear presentation of how you worked the model and integrate into the data. I was wondering if you thought about incorporating some of the uncertainty from the measurements. As you probably know, there's quite a bit of variability that's coming from the eddy covariance method itself. And these data sets that you use, they have some of this information. I was wondering if it would make sense to incorporate that as kind of a component of that uncertainty estimates that you are doing.
Yes, that's a good point. And I didn't bring it up, but we do. So when we calibrate the model with observations, we do need to provide some estimate of what that observed uncertainty is. And so with these, I think we were using monthly data. And I think there's a monthly uncertainty that's part of the standard data product. And so we use that, but it doesn't. So typically the model errors that we see are quite a bit larger than those when we do the calibrations.
Other questions? Thank you. Thank you so much. I really enjoyed your talk. I was very intrigued by the calibration results that you were showing. And those made me curious whether or not you were able to take a look at cross-validation results. So like a leave-out study, you train on a fraction of your data and then you evaluate on another fraction. Have you had a chance to take a look at something like that? And if so, how does performance stack up there?
Yeah, so we can do that in multiple ways. At an individual site, we can use just part of the record for calibration and then save a few years for validation. It tends to, so the uncertainties are, the errors are a little bit larger typically in the validation time series. But when we're trying to go from one site, so if we train at one site and then try to predict another site, then those errors get quite a bit larger. And so that's due to a lot of this variability that the model doesn't capture and other factors that the model doesn't consider maybe driving those differences in parameters.
Okay, we have time for one more.
Thank you for your talk. I think this was really interesting. And just going back to Gilberto's question, when you were incorporating the errors from the different sites, I was just wondering if you were on the monthly errors, if you were assuming heteroscedasticity or homoscedasticity.
For this, I think it's homoscedasticity. That's a hard one to say. But I don't know. So when you go from obviously the hourly measurements, that's a strong consideration, I think, because of the way that the eddy covariance method works. As we've looked at that for the high temporal frequency calibrations that I haven't done here. But I'd be interested in knowing how that persists at these longer time scales, whether or not that's necessary.
Oh, thank you.
Great. Thank you, Dan. I think we'll move to our next speaker. Thanks.
So our next speaker is Daniel Beverly from Indiana University. He's going to tell us something about his soil water retention curves. Thank you.
Talk. This is pretty awesome. We're going to talk about some of the soil water retention curve projects that we've been doing at IU. So you'll probably see this logo maybe at AGU or a couple other conferences, but yeah, so we've kind of termed this project the Regional Ecosystem Soil Hydraulics Project. And so before I get too deep into this, I just need to make sure I acknowledge all the funding sources and all the people who have helped either in the lab and creation of this project. And a lot of you have already contributed soils to our efforts into increasing soil water retention in AmeriFlux. So I really like to start with this figure here because it really highlights how complex of a systems that we are really interested in. So over the course of the last few days, we've really thought about how we're thinking about how can we understand and measure these different processes. If we think about soil, we started here at the soil water and soil water status. We can move through different levels in moving into the plant and how that interacts with CO2 and CO2 fixation, driving GPP and all these fluorescence parameters, ultimately increasing productivity of the ecosystem and respiration and backing into decomposition, and the whole process continues. I really want to talk about two primary components of this. Thinking about soil water content. We all measure it, almost all the sites do. And that really tells us a lot of the soil water status. But that really tells us the status of the soil and how much water is in the bucket. The other major, really important water status parameter is soil water potential. And water potential is a much more complicated measure to create because it's a much more integrative measure. When we think about soil water potential or bulk water potential, we think about the proxy of osmotic water potential, how much solutes are in there, how much energy is allowed to move between there. You have the pressure potential, or how much water or energy is on the column, pulling water from the soil through the plant, to the xylem, ultimately to the atmosphere. And then we have the matrix potential, which is essentially the interactions of the water and to the environment that it's in, whether that's in the xylem conduits or in a soil matrix. Gravimetric water potential, we don't usually consider too heavily in the soil world, but we do when we start thinking about soil plant interactions. And the real advantage of these two measures is when we can combine both how much water is in the system and how much energy is in the system to do work to help increase plant productivity and increase GPP and carbon fluxes. And this is where the issue really kind of begins. So we have an awesome network of sites. We have soil water content data available from all sorts of different sources, different frequencies, whether we're looking at mesonet sites across different ecosystems or different state levels. We have obviously we have AmeriFlux, FluxNet, Neon, all contributing to soil moisture data, where they're both different profiles and different sites. And we're even getting remote sensing soil moisture products for even larger landscapes. But we do not have soil water potential at these same measurement frequencies or same measurement locations. And there are very few sites that have in situ soil water potential sensors that are especially paired with soil moisture contents. Moreover, there's not very many sites that have site-specific soil water potential curves. So for example, here is the all in blue represents the all AmeriFlux sites based off of the website from a couple months ago, thinking about the soil texture derived from their spatial locations in the NADS soil texture maps. And then in Burgundy, we have the soils that our project or the REASH project that is trying to parameterize. So we're trying to cover most of the AmeriFlux soil moisture or soil texture domain. But since these sites do not have in situ sensors nor site-specific curves, this forces all of us to use texture-based petatransfer functions. And so when we think about texture-based petatransfer functions, we understand they're highly variable across any of these soil moisture or soil texture gradients, and how soil moisture and soil water interplays between these across different textures. And this becomes even more problematic if we think about how sensitive these functions are, especially since we're looking at this, these figures are soil moisture on the x-axis and water potential on the y-axis, and this is in the log space. So if we think about uncertainty, these are extreme uncertainties when we're just shifting subtle shift or making subtle shifts in the parameter of the Van Genoosen model. And if we think about how we use this data, and we're using these very, very coarse maps to estimate, soil water retention capacities across these ecosystems, this becomes even more complicated and more uncertain when we're really trying to constrain flux measurements. And this is a really fun example that I really enjoy from one of our sites in mid central Indiana. So this is a cornfield, but these are some of our soils in the cornfields where we have 10 centimeters. We have this super organic, rich black soils, and 50 centimeters right below in the same pit is almost pure clay. And we know the corn roots in these fields are playing or interacting with all these soil profiles, but our soil map suggests that it's a one parameter or one coefficient. So this driving both spatial and temporal or spatial and profile variation. And so to test some of these variations of both space, time, and across different ecosystems, we're going to focus on 3 different sites for today's talk. So we'll have a site in northern Michigan, that's the UMB site, the Morgan Monroe site up in Indiana, and then one of Dennis's cornfield sites in California. But so how do we actually characterize or fingerprint these soils? So we've been sending out soil collection kits. So some of you have either filled out the survey or received kits, and then we're able to go to your sites following our protocols, collect samples, ship them back to us. We prep them in the lab, catalog them, and then we start putting them into 2 different components of the soil water retention curve to derive your site-specific water retention curves. So we have our saturated water retention components, and then we have our unsaturated water retention components, and then we use those 2, combine them to derive essentially the fingerprint for that soil and that soil profile. Really, I need a shout out to make sure my technicians and everyone who has helped on this is being recognized, because they've put in significant hours in the lab making sure these curves are being made. But we're going to start with Michigan for this. And so on all these figures, you're going to see on the x-axis is going to be soil water content, matrix potential or soil water potential on the y-axis, and we're going to have our profiles. So we're going to have surface 10 centimeter, 50 centimeter, but we're also going to show how the pedotransfer function based off of soil texture fits and matches these models. So just looking at this figure, we see that there's a really, really unique shape at the University of Michigan. We have this extreme L characteristic of these soils. This is likely, or this is definitely due to the sandy nature of these soils. But the major concern that we can start with is the texture model is very, very off in this situation. Oops, wrong button. The other thing we can see is the texture model cannot account for any of the organic matter that's actually in this. It's solely based on sand, silt, clay content. But there is organic matter through the soil profile that's driving the soil or the saturated water retention components. So we have definitely variance in the soil profile, and we have very disagreement in the soil or in the pedotransfer function relationships relative to site-specific curves. Alternatively, we can move to a different soil. So Indiana soils, we have much siltier, loamier soils. And we can see that there's less variance and less L-shaped nature to these soils as we have a higher water retention capacity. And then we can see that the soil texture model is closer to some of these points. But the problem with this one is that the soil texture model is really close to the very low end of the water retention curve. And the soils in Indiana very rarely are below 20% volumetric water content. And if we look at the most organic rich, these organic, super high organic content soils are coming from the ag lands in California, we see that this linear relationship almost turns to, or this log linear relationship almost is linear, and we have significant less variation again in the depth profile. But the texture, once again, cannot account for the high saturated levels of these measures. So specifically, we want to really think that site-specific water retention curves are capturing this variation and this super important variation across both space, time. And then we are having, if we're relying on pedotransfer functions, we're going to have significant uncertainty across different sites and across different soil moisture regimes. But this still leaves a gap since these measurements are representative of a single point. How do we understand soil water potential across space and time? Especially when we think about how we link soil water potential and plant water potential. So when we think about how we measure soil water potential versus plant water potential, we can use this very basic space-time continuum. If we think that x-axis is time, and we have seconds to decades, we can have long continuous time series of soil moisture now. Now we have soil water retention curves or in situ sensors. And then we can think about how we do pressure chamber measurements and other plant water potentials, where we might take one plant water potential measurement a day, but across 15 trees or across a larger section of the landscape. So we're seeing this significant mismatch in both space and time. So is there a way that we can use both these soil water retention curves, in situ data, to expand our understanding and overlap water potential measurements with the plants? So the last bit of this talk, I'm just going to highlight some of the work we're doing in Morgan Monroe, linking electrical resistivity tomography measurements to soil plant water interactions. And so this is a lot driven by previous work that we've done, but we have had really good success, especially in more silty clay soil. So for example, this is an experiment that we were doing in Panama, where we ran this system for about 30 days. And you can clearly see where the water is being extracted across this hill slope gradient. So, and this is also based off similarly off of first principles, since Archie's law derives or links moisture content with the resistive or conductive nature of a material. So we can use this to generate very clean and process-based water calibrations in the lab that can be directly applied to the field. So for example, this is some of the water retention curves that we've just been playing with in the lab. So we collected the same kind of soils, and instead of using water potential or mass, we're using electrical resistivity to look at the relationship of water content versus electrical resistivity. And the really nice part is we can even clean this up significantly when we think about the relationship of electrical resistivity versus water potential, especially since electrical resistivity is sensitive to both matrix potential, osmotic potential, and all these other potential gradients that are driving moisture status in the soil. So I'm just going to show you one quick snapshot. So this is just a snapshot from late August in the Morgan Monroe State Forest. And we can see there's some trends going on. We're drier near where the plants should be. So the top is where the plants should be. We have 3 meters of our depth of investigation is about 3 meters. But if we put this into a little bit better context, where we actually map some of the trees and vegetation on the landscape, we start seeing, okay, bigger trees, deeper roots, more water uptake. So this is a snapshot. So this is not a percent change. So this is still very useful that we can see where water potential is greatest and the greatest differences across the thing. Lastly, if we zoom in, we can actually see where this water in this water potential gradient is actually forming. So we have essentially what we can deem as the root zone near the surface in the top meter, and it's essentially wicking water from the shallow or deeper wet rocks below the rooting zone. And this is just a time-lapse video. It's pretty grainy, but that's okay. But mostly we can see over time that the wet rock or the deeper profiles are getting drier and drier, and they're becoming smaller and smaller. So this mostly is just highlighting that electrical resistivity paired with some soil water retention curves provides a promising insight onto spatial and temporal variation in both water potential across much larger areas and deeper profiles than we previously have been measuring. So lastly, we're just going to end on some of our summary slides, thinking about what are our data products that we're trying to produce with these electrical resistivity and soil water retention curves. So we're trying to get more site-specific soil water retention curves for all of the AmeriFlux sites, specifically the core sites at the moment. And then can we link these to both different types of fluxes? Can we expand our water potential understanding both across space and time? To do this, we're using our site-specific water retention curves. We're looking for anyone who has profiled or in situ soil water retention curves. and then other sites that have electrical resistivity that are running on time-lapse series. So next, we're still thinking about soil water retention curves and how can we improve the models to actually account for the organic water or the organic matter content. Improving or getting better characterization of sandy soils, and then establishing more electrical resistivity lines across the landscape and across AmeriFlux sites. Quick. So, and network research coordination network to increase water potential data and address intractable questions about plant water status and flows. So our goals are to build extensive databases that are holding and collecting water potential data for both soils and plants on cross time series scales. Right now, we're starting to collect pressure chamber data. If you scan this QR code, it'll take you to the website where we can start do pre-surveys on entering data. We're working on best practices for measurements, both pressure chamber and psychrometers or other in situ sensors. We're doing FizFest is next week, I believe, or 2 weeks maybe, but there's other graduate seminars and other research opportunities. And of course, we'll be present at AmeriFlux with different workshops and networking activities. And lastly, if you're interested in joining or contributing soils to this project, we would love to have you scan and fill out this QR code. We'll take you to our pre-survey. They'll ask a bunch of questions. We'll send you a sampling kit and essentially return soils, and we'll return data and a bunch of curbs and all that. So with that, I'll gladly field any questions.
Time for one quick question for Dan. Okay, we'll move on to, yeah, a little bit behind time. We'll move on. Our next talk is a team talk from Angela Lafuente from Michigan Tech University. Tyler Roman will be joining from USDA, and John Rengifo from the Amazonian Institute. He's down in Iquitos.
Is this the correct view, or are you seeing the presenter view?
Presenter. It is the model view, not the presenter.
That's better.
Yeah.
Okay.
Yeah, it's better now.
Okay, go ahead, John.
Okay. Hello, everyone. Here we are, Angela Lafuente, Tyler Roman, and me, John Rengifo. We are going to present what we found contrasting with the dry season carbon dioxide and methane fluxes of the Amazon peatlands. Next slide. I will start saying that the peatlands are the very important ecosystem. They cover about 3% of the Earth's surface, but they can store twice as much carbon that the world forest and are crucial to mitigate the climate change. A group of these are found in the tropical zone, which we don't understand better and are understudied, but all our efforts are concentrated on it. The Loreto region in the Amazon, in the Peru Amazon, contains about 63,000 square kilometers of peatlands, and it's where our study site is located, just a few kilometers from Iquitos City. In this slide, we show the aguajal of the Quistococha. The aguajal is a swampland ecosystem dominated by aguaje, the palm tree, Mauritia flexosa vegetal species. This ecosystem is important because it has an average of 2 meters of peat that can store about 740 tons of carbon per hectare in the soil. So in 2016, with the main goals of to better understanding how the aguajal works regarding to greenhouse gases cycles, a tower was established with implementation that measure the carbon dioxide and methane fluxes at ecosystem level using eddy covariance methodology. In addition to this, in 2021, an automatic chamber-based system using LICOR instrumentation was installed in a circular plot of 15-meter radius within the footprint of the tower and containing the most abundant plant species of the site. To measure carbon dioxide and methane fluxes at sub-ecosystem level, trying to understand how these components, the soil and stem fluxes contribute to overall the ecosystem fluxes. 8 soil chambers were installed covering the spatial variability of the site, and on 8 selected plants, stem chambers were installed at 50 centimeter height, which were constantly measured. Within the plot, there are wells with sensors that measure the depth of the water table to understand how the ecosystem responds to the dry and wet season. Now, my colleagues will explain some data that we found in the next slide.
And we will start focusing on the fluxes from the chamber system. In this figure, we see the temporal series of all the methane fluxes recorded from soils in red, together with the variation on the water table in blue. We identified 4 drying periods, and we can see a temporal variability in the soil methane emissions. I want to point out the reductions in soil methane emissions during those drying periods, and it's especially clear during the drought that we found between September and October 22. If we look at the relationship between the water table and the soil methane fluxes, there is a clear relationship. When soils are dry, methane emissions are very low, and they increase with increasing water table up to about 15 centimeters above the soil surface, like the water table, when we can see a threshold. And after that threshold, soil emissions decrease. When we look at the stem fluxes, we have measured Mauritia flexuosa, which is the dominant palm tree, Mauritiela, another palm tree, and Tabebuya, a tree representative. And we see a high temporal variability on these stem fluxes. Again, when we compare the relationship between the stem fluxes and the water table, we see that fluxes vary by species. In yesterday's talk, Rodrigo Vargas gave a talk on AmeriFlux about the stem fluxes, and following on the interest that the stem fluxes has on the scientific community, we need to upscale those stem fluxes in order to understand the contribution that trees have on the ecosystem. So we use the vertical changes on methane fluxes. We measure different heights on different trees. We use the stem surface area and the tree density on the site to upscale those stem fluxes that we have measured. And we can see differences on the contribution of the different species depending on the water table. Mauritia stems have a similar relationship to water table as soils. They saw a similar threshold between 10 and 15 centimeters, whereas Tabebuya's methane fluxes peak when soils are very wet. So following on the contribution of soils and stems, and based on our calculations, stem fluxes only represent less than 1.5% of the total fluxes. Next. Yeah, this is our first attempt to put together the data from the chamber system and the eddy covariance tower, but bringing both together, we still have lots to learn and too many questions. But because of what I've explained, that the stem fluxes are such a small fraction of the ecosystem based on our calculations, we are only showing soil fluxes in red together with the eddy covariance methane fluxes in gray. Our results are in agreement with the literature. We see in general higher methane emissions from soil chamber system than from the eddy covariance. And we see a similar response between soils and eddy covariance. However, the response varies when soils are very wet. And as I said, we still have lots to learn in here.
And so turning to the carbon dioxide side of things, here we see the time series of the soil chamber fluxes measured throughout this study period. And it is important to note these are opaque chambers, so this should be respiration only that we're seeing. And again, during the dry periods, and particularly in October and January, as that water table is dropping and the soil is drying out, we see a pretty large increase in the CO2 flux measured in the soil chambers. And so if we look at that as the flux related to the water table, we see that that increase starts to happen once the water table drops below about 10 centimeters or so above the surface. And then that flux rate continues to increase as conditions get drier. And then if we contrast that with the stem fluxes, we see the CO2 flux from the different species, the rate varies by species, and there is some relation to water table, although not nearly as prominent as the soil-based chambers. But we do see that the palm species have a similar trend where we see a little bit of increase as the water table drops, but again, not as prominent as the soil chambers. And so then bringing together, again, the tower-based data and the chamber-based data, here what we're showing from the tower is just nighttime NEE to represent respiration. So this isn't results from partitioning or anything. But we see when we look at the dry periods, again, focusing on that window in October, the tower-based fluxes do show a bit of an increase in the respiration rates during that dry period. And during the wetter seasons, we see the eddy covariance shows a higher respiration rate overall, but then in that drier period, we start to see some overlap between the soil chambers and the eddy covariance system. And there's likely multiple factors at play here. One would just be on the physical side of things. As the soil is drying out and the water table is dropping, it's easier for gas just to diffuse through the surface compared to when it's waterlogged. So some of that might be reaching our instruments better during the dry periods. And then there's likely some sort of biological component here as well. During that dry period, maybe what we're seeing at the ecosystem level from the tower is driven more by soil chambers versus during the wet season, maybe there's more vegetative respiration that's being captured. And so some of our summary here is that we do see at both the eddy covariance and the chamber system level, we see correlation between water table and both the methane and CO2 fluxes, and those responses vary based on different factors. And overall, the flux rates on the stems do vary by species. And when you do the upscaling, as we've done for the methane flux, the stems represent a very small proportion of the overall flux when compared with the soil chambers. And so moving forward, there's a lot more that we can do to better understand this ecosystem. So part of that will be continuing this investigation of the vertical distribution of fluxes, both along the stems from the soil surface upwards, but then also utilizing profiles data that we now have on the tower to get a better understanding of what's happening from the soil surface up through the canopy and to the level of the tower instrumentation at the top of the tower. And then lastly, a lot of the data we've shown here have been collected largely during La Nina periods, and this area is forecasted to go into an El Nino, so drier, hotter conditions moving forward. And this figure down here at the bottom is our water table updated as of this Monday. And you can see that right now we're entering another one of these dry periods, which should really help to expand our data set to gain a better handle on what's happening here. And then a quick plug for N2O fluxes at the site are coming soon. Our colleagues at the University of Tartu in Estonia were granted funding to add some N2O instrumentation to the site. So chamber-based instruments were added in March of this year, and then eddy covariance instruments are coming in the coming months. So keep an eye out for results on that front in the future. And then lastly, just thanks to everybody that made this possible, all these different organizations involved, our funders, and then a special shout out to the AmeriFlux tech team and the instrumentation loan program, which was crucial for keeping our site operational while we dealt with some instrumentation damage and repairs. And so with that, we thank you for your time, and we'll take questions if we have time for any.
We have time for about 2 questions.
There's a that is, whether it's to the full height of the tree, whether it includes branches, how high up the measurements went. We're working on similar things in temperate forest, and those make huge differences in, you know, what the actual emitting surface area is. So just wondering how you approached that.
I'm not sure if it was just me, but the audio broke up there at the beginning of your comment. If you could just repeat what you said at the beginning, that would be helpful.
Is this coming through? Okay.
Yes, better now.
Okay. Yeah, I was just wondering if you could say more about how you scaled stem fluxes to what height, to which surfaces of the tree, to what total areas, and how you got those estimates.
Yeah, so we have these automatic chambers. We have been measured at 2 different heights, 50 centimeters and 90 centimeters for over a year. And then we did 3 or 4 field campaigns where we took manual measurements at different heights. We went as low as we could in the tree and up to about 2 meters or 230, I think was the maximum height. And we observed a clear pattern in all the trees that we measure, where we had very small fluxes at the bottom. We saw a peak on the fluxes, and then the fluxes decreased to almost nothing. Over, it was like above 1 meter on the tabebuyas. Fluxes were almost 0, and Mauritius kept the fluxes a bit higher than that, but at 2 meters, all the fluxes were almost 0. So based on that, we have assumed that trees are a cylinder, like tabebuyas are considered like a cylinder up to 2 meters. And for Mauritius, we went a bit higher. We consider like, okay, it's a cylinder up to 5 meters. And we calculate the flux, like the surface of the tree as a cylinder from 0 to 5 meters. And I fit an equation based on the patterns that we observe on height on the trees. Does this answer the question?
Time for one more question. Well, I do have a question, Angela. So I'm curious, in some studies, the stem flux was large and it seemed to reconcile the difference between top-down and bottom-up methane emission estimates. So I'm wondering if you have a hypothesis for why the stem fluxes are so small at your site.
We're still thinking. I've been reading the literature, and there are different ways people have been calculating how to upscale the fluxes. Many research, what they have done is assume it's a cylinder and assume that fluxes are constant, which that really overestimates, because what we have seen is like up 2 meters, the fluxes are almost 0. And many times it's like, well, we will measure one height and assume that the tree is 10 meters tall and fluxes are the same at 50 centimeters, which are higher than another height. And then go like, oh, that's the same for the whole tree. So that could be one of the explanations, that we measure different heights, we saw a different pattern, and we fit an equation to how the fluxes respond to the trees on this area. Why the trees have that pattern, we haven't done any sap flow measurements yet, or any bark measurements on density and how the flow could be, if it's related to the water table. We don't know that yet.
One more question.
This is Rodrigo Vargas. Just a brief comment. We can take this offline, but might be that you want to look a little bit more about the wood characteristics. I don't know if these are just palms, and if they are palms, maybe they are just sucking the methane from the soils and just pumping out in the very top. You know, because of how the palms are. If you cut a palm, you don't have all these the structures they have. So maybe you might be measuring the methane fluxes in the wrong place, because in soils, because of this heterogeneity that you have a drought, and now you have a more agreement between the ecovariance and the soils, but then you have the water, and then everything just like lights up, and you have heterogeneity. Same things with the trees. It might be that you're just measuring where there are low fluxes, but the high fluxes maybe in the very top, and it's this complexity, but I'm happy to chat in more detail after that.
Yeah. Yeah, thank you very much.
Thank you. We'll move on to our next talk.
Our next speaker is David Reed from Yale University. He's going to tell us something about continental scale carbon fluxes.
Hopefully I will. We will see. Okay. Good morning, friends. Breaks are important, and so we are a little bit behind schedule. And hopefully I will get very excited and talk about this very quickly and maybe give us a couple minutes break before the second morning session. Yeah, let's just go into this. There's just a delay there, isn't there? Okay, getting used to this. Lots of us, particularly the American AmeriFlux management program folks, have shown some version of this figure. This is a couple months old, but there's lots of AmeriFlux sites in the network. We often think of these as little point sources of data, and it came out in the ecological breakout session of Dave Moore's. Like, my point here, putting on an ecological hat, is that our data is very cool and unique and powerful. And one of the things I'm always really interested in is how much we can kind of infer across the landscape from these point sources. And so I'm always inspired coming to these meetings because you guys do all this really cool and innovative and complex work. I feel like my work is much more simple and straightforward, but it's a lot of the same stuff, right? Can you take eddy covariance data and build this very complicated model and then use that to infer our information across the landscape? Can you do upscaling, downscaling, all this other really cool work? So real quick, again, we're going to go through kind of a conceptual overview here. If we have a couple sites across a cluster or a little bit of a landscape here, how can we apply information between sites or maybe across sites or across the rest of the landscape again? So just to kind of walk us through some of this, I ended up taking some of the data from Jiquan Chen from our old Michigan cluster. Some of these sites are no longer active, right? But we have information on these sites. They're not just red dots in the landscape. And so if we put what type of land cover information there, automatically, a lot of you guys are starting to think about, well, what can we do here, right? And so we have actual data, and I'm super glad we have more and more people doing urban fluxes because it's super important and also super hard. And I feel less bad that my urban data is super bad because everybody's urban data is just different and unique. Right. And again, Jiquan's not here. So I will tell all of you guys, there's a massive amount of agricultural data that Jiquan has. And so if you are on Zoom and you can actually like see some of this data because it's small here, if you squint into this, some of these agriculture sites have like 13, 14, 15 years of data. And then again, some of the newer sites have 3. But ultimately, like, I want to try to compare some of these. So corn and corn, switchgrass and switchgrass makes sense. If you're doing corn work, you can use corn data from the other side of the county, or in this case, for some of Jiquan's data, like just down the road or across the road, to do whatever you need to do. Gap fill your data, help parameterize, run comparisons. For this work, these paired sites all have very different land use histories, but current management practices. And so there's really cool things we can kind of pull from this data. And again, we have. But again, let's start taking a step back, right? If I didn't tell you what these sites were, and I just show you the data, we could do some fun active learning exercise where I'd give you all the data and you can zoom in and you could try to guess what type of land cover types these are. And I think you could probably do a pretty good job, right? Because we kind of intuitively know what some of this data looks like, at least the urban one. I think we all could be like, this is either noise or bad data or urban data. It's one of the three. But once we start doing this kind of exercise of like removing ourselves from the picture and some of our biases and just let the data show it, like what information is there, I think that's my starting point. We can start doing some cool stuff, right? We can start comparing. And we know some of these comparisons are going to be good. Some are going to be bad. That's the point of doing it. So again, let's just get rid of the data then, right? The data is going to come into this in just a second, but let's just start comparing sites across the landscape to see which ones are close to each other, which ones are not. Again, we'll get to some useful implications of this in a second, but we can do this for all possible sites. And so sadly, because of how presentation schedules sometimes work, if you guys saw some of this setup, maybe it would have made my students Savannah's poster a little bit more sense, because she was showing data from not individual sites, but comparing all possible site pairs across the landscape. And that's kind of a weird thing if you were just to see it on a poster. But that's where we're going with this, right? So how to actually do this. Now I'm going to point to my other co-author, Hausen. This is all of his work and his code, and I've just kind of adopted it and used it to kind of do some fancy graphing with it. But let's take, they are labeled up there, corn and urban, right? The most different sites possible. Again, the corn has 13 or 14 years of data. The urban has 3 very gappy years. How do we compare these? I don't know, but Hausen does. So the first thing you have to do real quickly is how do you, again, characterize ideally a very long data set? And so some of you guys have seen this already, and we've kind of went back and kind of looked at some different ways of doing this exact kind of time series characterization work, because you guys gave us very insightful comments. But what Hausen has done here is taken, again, very long data sets and created kind of a characteristic or an average 1 year out of it with different varying kind of windows within it. So on the far left, we have 5-day windows, and within each one of those representative day windows, we have 3-hour data points, and then a 1-week window across that year with more daily temporal resolution, and finally a larger window of time with less daily more daily resolution. Ultimately trying to, again, take large data sets, pull out the important information from it, leave behind some of the noise, and yes, arguably some of the variability that's important, but get a characteristic time series from each one of these sites. We found out that it doesn't really change results that much. And so right now we're kind of focused on the midpoint there. So we have a characteristic time series of corn that looks exactly what you would expect from corn, right? It just is ripping during the summer. It has a fairly short growing season. And then we have, again, urban data that just looks like MATLAB vomited up some data for all of us. But the cool thing is, right, is we can actually use this to compare these two results. And again, corn and urban are not going to be similar. And so I'm about to show you a number right here that is not a good number. And what this is basically just the calculus here between these two time series. How different are these two time series are from each other? The more different they are, the higher the number. You're just kind of like integrating along the time series and adding up all of the errors, but I don't like that because that's a negative word. You're just adding up all the differences. And so between corn and urban, it's a number. It's a high number. And what I end up doing anyway is normalizing all between 0 and 1. So this is just a score of how similar these are, or how dissimilar they are to each other. And then just looking at these raw numbers, right? Like corn and the forest site that I'm adding in here, so we have more than just one number to talk about and compare to, is a different number, and it's less, and it's closer to each other. And just by eye, they're not similar, but they are closer to each other, right? You still have a large photosynthesis respiration signal in the middle of growing season. The forest is a much larger growing season window, but we can, again, the point here is quantify how similar or dissimilar any two site pairs across the entire network is. Okay, gotta take a breath here. Again, excited, talking fast, trying to get us to a break. Some of you guys have seen the first version of this manuscript. You just ignore me and my promises that I make. You're going to see another version of this manuscript in a couple weeks. It may be a month or two, but before AGU for sure. I want to pivot really quickly and say I really want my work to be as inclusive as possible. The next manuscript to include them on the authorship Google form, please. I've run sites myself. It's exhausting. I can't do cool work like this, except in that Michigan cluster of sites, maybe, but I can't do it at continental scales without all of our data. And so like, if you have other people you want to add to this paper, and then the next one I'll show you in a couple slides, please do. So I'll send that out in an email. Again, you've seen some of this. What we can do with these site pair differences then is compare them, cool, and then also start to cluster the sites. And so I have to remember my own. This is air temperature. I always go back and forth between air temperature and incoming solar radiation because they cluster out so well and exactly what you'd expect, right? You have a couple sites on the north slope of Alaska that cluster out because air temperature on the north slope of Alaska is not the same as the rest of Alaska and the rest of the boreals. You have, our tropical sites are kind of clustering out. This looks right. It looks like everything's working well enough. Here is NEE. And it's a lot more complicated because I would say, and I hope most of us would agree here, and hopefully the reviewers agree later on, is that NEE is a lot more complicated signal. There's something in here. There's a reason why some of these clusters are kind of popping out. And I can't fully explain it. We may never be able to fully explain it, but there's a lot of interesting stuff in here that could be useful to our community, or maybe it's just a cool map to see. Yeah, so again, trying to rush through, get to break. If you have comments or questions on this, let's chat afterwards. But right, one of the things we can do to start to pivot is also look at, again, we have gone back and redone this, but this is a pretty map, and I want to include the pretty map and not necessarily like the work in progress map for this, is combined all of the Flux data together and get a one metric of site uniqueness. And so again, this to me clusters out really well, I think, where you have, again, some, Everglades sites and North Slope of Alaska sites that just look different from the rest of the network. And I think that that's important. And I also want to say some of the sites that look very similar to each other, depending on what your needs are, right? If you need a lot of agriculture data and you want it to be very similar with small differences, this is probably a good way of justifying why I'm pulling in a bunch of corn data from like many miles away. Okay, so let me pivot really quickly to some very preliminary results on kind of a follow-up paper here that I'm working on that, again, feel free to ignore this. My promise is I hope to have a draft out to potential co-authors before AGU, but I'm lying. It'll get done when it gets done. We can also go back to this inter-site comparison across the network idea and look at spatially how far these sites are, right? Again, I'm not putting in any human biases in here. This is just a GPS metadata point and then a bit of code to give me all of these potential site differences, spatial distances across the landscape, and compare. So this is NEE. And I was really struggling with a way to kind of frame this in an interesting way. Then, again, it came out in the ecology breakout group. Like, this is what ecologists do. We take, putting on an ecology hat here, we take data and we try to scale it across the landscape to see if I'm measuring NEE, how valid it is outside of this home forest, right? If I'm collecting NEE in one forest, if I look across the valley to the next forest over there, are any of my measures here scalable over there? And I am going to come back later and kind of break this down by land cover type and kind of really dig into this a lot more, bring in, right now I'm just going to show you the next two slides, just carbon fluxes, but I'll look at all the other fluxes as well here. But it's very similar. So what again, I hinted at it before. What I've done here is just take those large numbers, hundreds or 200s, similar or dissimilar scores, and normalize it. So we have 0 is completely dissimilar, 1 is very similar. And again, this is all possible site pairs across the network. So we go from 400 and something or other sites to thousands of potential site pairs or tens of thousands. So it's a huge cloud of data. And again, like I've looked at it really quickly, but it's not included in this. It does break down very well between different land cover types. If you're looking at this just for agriculture, it's a different relationship for wetlands. But what this is hinting at here is that our sites are very similar as we go across the landscape. So if we're measuring NEE at Harvard Forest over there, we maybe don't need to set up another site over here without a very good reason, right? Harvard Forest has the hemlock site and the neon site right there, and they're kind of measuring different things, and that's important. We don't have perfect similarity, but there's still a lot of power and information in that data from Harvard Forest. And if you were talking to a forestry colleague that had plots just over there, and you wanted to have a conversation about, well, can I take GPP data and give it to you? Maybe you can come to a very reasonable conclusion that says, well, I'm not measuring GPP over here. You're measuring your DBH or your forestry or whatever it is. Maybe it's your deer grazing, deer exclusion plots that some forest ecologists have. And you say, well, okay, well, I can't move my tower over there and you can't move your work over to the Harvard Forest. Well, sorry, we can't really collaborate. I think we can. I think a lot of our information can scale spatially. And so we can maybe use some of this to justify kind of like talking with our colleagues down the hall and say, cool, you have sites in a similar ecosystem that's spatially far away. We can still compare our stuff. Remember, eddy covariance data is not objective measurement of truth, and there's error involved there. And so there's error involved with kind of scaling our information across the landscape that may be the same exact, order of magnitude error of just our measurements from our sites ourselves. So that's NEE at the top here. I also have GPP respiration first and then GPP. And a little bit surprising to me is that the photosynthesis relationship kind of drops off faster. I would have thought it would have been the exact opposite, but it looks like this here across all sites in the network. Respiration scales out across the landscape pretty darn well. And if you really want to squint here, like these are thousands of kilometers here. And so if you're taking respiration measurements at a forest, it's actually kind of sort of similar to respiration at a cornfield if you're close together. Respiration at a forest is pretty similar to respiration in another forest, even though, again, you could be pretty far apart. More to do here. Last slide before break. Also, housing did all the work. I just plotted it up. But we have methane here. Methane, let me just put a caveat in here, is different. Methane does not scale. Methane sites are unique. We all know that. And we've had talks here from we need a little bit more representativeness of methane. We need more methane sites. Methane probably doesn't scale very well. And that's cool and that's interesting. I don't know exactly how to handle that right now, other than just kind of a plea back out to the community that let's kind of keep working on methane. But there doesn't seem to be a relationship across space with methane. So thank you. Let's go have a break. If you have questions, talk to me during the break or right now.
Thanks, David. Time for one question.
Thank you. Very interesting talk. Well, all the sites are under the same sun on the same earth. That itself should already impose a lot of similarities among different sites. But I wonder, the score you calculated between sites, are they really comparable, or do they give a certain sense of, or if you have similar scores between two sides, and then they are compared with another pair have similar scores, do they really comparable with each other between the two sets of two sides?
I think I understand what you're asking. Everything's comparable, right? It may not be a good comparison.
Well, if you go back to that, you have three sides, triangles. Yeah.
Yeah.
So the corn and the forest, it seems to be the similarity or the difference between them should be much smaller than the size between urban and a corn and an urban and a forest. But the score doesn't seem to be very much different. So I'm just wondering.
This is just math land, right? Like these are non-dimensional comparisons here. One way of looking at this is that's, you know, what is that, a 40% reduction in whatever that term is. Obviously what I should have done if I was a little bit smarter and forward thinking is have that second corn site there. And I think the number would be like 10 or 20 or something between two different corn sites that are very similar. I just wanted to show this up here to show that corn and a forest site are a lot closer than corn and urban and forest and urban, and make the point that, you know, the urban data we know is not going to be reflective of kind of terrestrial ecology sites. And sure enough, that is. Does that make sense? So yes, some of these sites, if I would have been a little bit more forward thinking and picking out the representative ones there, are a lot closer, because in fact, two corn sites that are spatially like 4 miles apart are in fact very close to each other. But I'm not that smart to show that. So again, break time, Tim?
We're going to have a 5-minute coffee break.
Okay.
Thank you, everyone.
We have a team talk from Andrew Richardson at Northern Arizona University and Adam Young with Neon, and they're going to be telling us about the links between phenology with their phenocam work and land-atmosphere interactions.
My name is Andrew Richardson. I'm a professor at Northern Arizona University in Flagstaff, Arizona. I've also been co-PI at the Bartlett Experimental Forest AmeriFlux site since 2004. And since last fall, I've been co-chair of the AmeriFlux Science Steering Committee. I'm sorry I can't be there in person this week, but I appreciate the opportunity to present this talk virtually. So today I'm going to tell you about tracking vegetation phenology using phenocam data and imagery, which I think is a topic that really nicely fits in with the AmeriFlux theme here of remote sensing. My talk is going to be pretty quick, and then in the next presentation, Adam Young is going to tell you more about how he's been using phenocam imagery to integrate phenology and tower flux measurements. So phenocams are basically just digital cameras that are used to track vegetation phenology using repeat photography. And we've been using phenocams to link across different scales from leaf level as seen by a human observer to ecosystem level as seen by satellite sensors orbiting the Earth. And then we also have used these data to facilitate analyzing connections between ecosystem phenology and ecosystem processes related to the exchanges of carbon, water, and energy as measured with tower-mounted flux instruments. And this is really giving us new insight into the sensitivity of phenology to variation in environmental factors, and also of the role of phenology in regulating the seasonality of how ecosystems work. Now, a really neat thing about phenocam images is that we can extract quantitative data from them. And this is made possible because digital camera images use what is known as the red, green, blue additive color model. And that model says that we can reproduce any color as perceived by the human eye through some combination of these three primary colors, red, green, and blue. And so that means we can take an image and we can split it into the three components. It's red, green, and blue channels. And then for a particular region, denoted by this yellow box, within each image, we can quantify the intensity or brightness of each of those three color channels. And that gives us a quantitative measure of canopy color, from which we can derive a quantitative index of canopy greenness. And our preferred metric is the green chromatic coordinate, or GCC, which is basically just the ratio of the intensity of the green channel to the sum of the red, green, and blue channels. And the graph here across the bottom just shows a dozen years of canopy GCC data derived from imagery from the Harvard Forest PhenoCam that I first set up in 2008. You can see that greenness is low here in the wintertime. It rises steeply in the spring. It drops off gradually over the summer, and it falls sharply in autumn. And then these patterns repeat themselves year after year. Now, transition dates that we can extract from inflection points in these time series correspond really nicely to dates of leaf out, peak canopy development, and autumn coloration as recorded by an observer based on their visual assessment of phenology. So our camera-based method is providing data that is consistent with ground observations, but automatically and at a much higher temporal resolution. And this same approach works really nicely in other ecosystems, from grasslands to agricultural crops, as well as shrublands and even evergreen forests. And this graph is also showing data from Harvard Forest. You can see that the GCC index is tracking the phenology of photosynthesis pretty well. So the green line is our camera GCC, and the blue dots are daily GPP derived from the tower flux measurements. And this is one of the main ways in which people are using PhenoCam data to understand how seasonal variation in vegetation activity is related to carbon and water fluxes at the ecosystem scale as measured by at ecovariance. So the PhenoCam network, which I run, evolved from a single camera, which we set up at Bartlett Experimental Forest in 2005. There's now almost 700 cameras, active cameras contributing imagery to the network. They all follow a common protocol. And so this is truly giving us a continental scale phenological observatory. North American coverage goes from Alaska to Hawaii and from Puerto Rico to Maine, with most EPA level 2 ecoregions having multiple cameras in them. And we're planning on the next curated data release, version 3 of the PhenoCam data set, coming soon. It's going to have about 5,000 site years of data and imagery. Within PhenoCam, we have excellent representation of AmeriFlux sites. There are 177 active PhenoCam sites that identify with AmeriFlux. Some of the AmeriFlux sites have had cameras for over 10 years. And there's really there's ongoing efforts between the AmeriFlux Management Project and my team to promote integration between our web pages and databases to improve data discoverability and interoperability. Now, all the data and imagery from PhenoCam are publicly available in near real time through our project webpage, which is phenocam.nau.edu. We've got a number of R packages, including the PhenoCam API R package, which facilitates data access and analysis. And we have a data access tutorial and many other resources available on our webpage. I just want to wrap up by highlighting some of the science questions that my group and actually many other groups are addressing using PhenoCam data, often in conjunction with tower measured fluxes. So questions like, how does vegetation phenology regulate the seasonal rhythms of carbon and water fluxes? What are the environmental drivers of phenology in different ecosystem types? How do climate change and climate variability influence phenology? How will phenology change over the next 50 to 100 years? And what are the implications of these shifts for ecosystem services related to carbon and water? Just to give you a preview of some things coming soon, you can look for a review paper coming out in AFM before too long, I hope, and also the data descriptor for the version 3 data set. It's in prep for Earth System Science data. Thank you for listening, and if you have any questions, please reach out to me or to phenocam@nau.edu. Thank you very much.
Hi, can you all hear me? Yes, great. Thank you.
Yes, we can hear you.
All right, thank you. Hi, I'm Adam. I'm an environmental scientist working with the Surface Atmosphere Exchange Group at NEON. And I also want to thank you for giving me the chance to share some of my work with you today. And like Andrew, I'm sorry I could not present this in person. So today I'm going to share work that I did while I was a postdoc working at Northern Arizona University. This work was primarily funded by an NSF Macrosystems Biology Award, and I've listed the collaborators who worked on us through this grant below. And I also want to thank and acknowledge NEON for providing the support that allowed me to prepare and give this talk today. So Andrew gave a really great overview and introduction to what PhenoCam is, the kind of data sets or derived products it can provide regarding studying phenology, and some of the applications or scientific questions that can be addressed using PhenoCam. And so what I want to do today with the few minutes I have is build on Andrew's talk, add to it, and give some specific examples of how I've used PhenoCam in my work to study how phenology influences fluxes of heat and water in different ecosystems across the AmeriFlux network. And here we just have some simple time series showing variability in both sensible and latent heat fluxes and how they line up with patterns of vegetation greenness from PhenoCam or phenology. For my first example, I'm going to share results or work we did that looked at how phenology influences seasonal patterns or shifts in sensible heat flux. For this work, we used what's called a big leaf modeling framework. And within this framework, sensible heat is characterized as primarily between the difference between the surface and the atmospheric temperatures, which is then modified by this aerodynamic resistance term in the denominator. This resistance term can be further broken down into resistances to momentum and an excess or boundary layer resistance term. And within this framework, the other kind of key components I want to mention or highlight are these two roughness lengths. The first one being the roughness length momentum, which characterizes or defines the height at which the wind profile extrapolates to zero. And then a roughness length for heat exchange, which is an empirical source or empirical value that defines the source height of heat in the temperature profile from the surface to the atmosphere. And so using this kind of big leaf modeling approach where we consider the ecosystem to be a single layer with no real vertical variation from the soil through the canopy, our first attempt was to use AmeriFlux data to try to see if we could predict the full seasonal pattern of sensible heat flux at different sites. Doing this, we initially found we were getting some pretty interesting results, yet they were slightly problematic given our goals. Specifically, we can look at this plot here. We can see what we were predicting in yellow, what was being observed or measured at the flux tower in red. And then, of course, we have our greenness curve from PhenoCam on there as well. Essentially, what we see are these pretty big differences between what we're predicting and what's being measured. But these over predictions or these extreme biases are not random or uniform through time. They have a really strong seasonal cycle or pattern to them. And specifically, we see that they're occurring during these phenological transition periods, either during green up in the spring or senescence in the fall. Digging into this model and kind of all its underlying components, we eventually determined that this resistance term was what's driving these large errors during these phenological transitions. And more specifically, it was due to the difference between these two roughness lengths over the course of the year or between different seasons. And so the difference between these two roughness lengths for both momentum and heat is characterized by what's called the KB inverse parameter or term. And why that's important is because it's a key kind of component of this boundary layer or excess resistance term. And so as the difference between these two values increases or decreases over the course of the year, the resistance likewise will increase or decrease. And in the types of models we were using or how we were parameterizing resistance in our model, we essentially were assuming a lot less resistance during this time of year, leading to a significant overprediction of sensible heat flux. We ultimately were able to kind of determine this and figure this out by backing out and estimating this KB inverse value for a bunch of different sites within the AmeriFlux network, and then plotting their time series, and then comparing that to what we get from phenology using PhenoCam. So here we have our KB inverse term, and we see that it's higher in the dormant season, and then it decreases as we go into the growing season, which is inversely related to greenness over the course of the year. And then I've also identified the phenocam transition dates identified as reaching 10% of the total seasonal amplitude. And the day of year is just up here. So this would be day of year 100. And we can look at 2013, for example, and we see as we cross or we start to see this green up, that's when we start to see a decrease in KB inverse as well. And to maybe kind of confirm that this is indeed at least partially linked to phenology, we can look at 2012 as an example, where green up occurred almost a whole month earlier relative to 2013 due to a warmer spring. And again, we see that decrease or that shift in KB inverse occurring right when we start to see that green up. We expanded this to 23 sites across the AmeriFlux network. And what we did is we identified transition dates for KB inverse, or that difference between those two roughness lengths. And then we compared that to the phenocam derived green updates. And what we see is that there's a really tight alignment in certain ecosystems with certain plant functional types, like in deciduous broadleaf forests or grasslands, for example. Well, in other types of ecosystems like those characterized by evergreen needle-leaf forests, we don't see that alignment at all, which is indicated by these green squares here. And so this and a lot of other kind of related research was published in a paper in Agricultural Forest Meteorology a few years ago. But the main kind of take home here for this talk is just I wanted to provide an explicit example regarding how we used phenocam-derived transition dates and use that in combination with different AmeriFlux data to better understand how we model and predict sensible heat flux across a wide range of ecosystems. Within the last couple of minutes here, I want to give a really brief overview of another study we did using phenocam to study seasonal patterns, in this case regarding evapotranspiration. So in this study, what we did is we were interested in better understanding what are the primary or most important drivers of seasonal patterns of evapotranspiration in different ecosystems. And specifically, we were interested in better comparing systems that occur in more arid regions, like in the Southwest, versus wetter regions, like in the Northeast. And we defined an aridity index defined as the 30-year average potential evapotranspiration divided by precipitation. And we selected 20 different AmeriFlux sites that broadly span this gradient. And so we modeled the primary drivers within for your seasonal evapotranspiration within each one of these sites. And then we were able to evaluate it along this entire gradient to better identify what were the primary drivers of seasonal evapotranspiration, and ultimately, how does phenology vary in its role or importance. To do this, we used a method called path analysis, which is very similar to structural equation modeling. And so this is essentially just a fancy regression model, or it's a framework that effectively links multiple individual regression models together. And why we chose that is because it allowed us to calculate both the direct and indirect effects of a given variable on, in this case, we're using bulk surface conductance because it's strongly related to evapotranspiration. And so for example, we can look at the direct effect of precipitation on bulk surface conductance directly, and then as well as it's mediated by GCC, or that phenocam greenness index. So we ran this type of model using time series for the whole year, or I'm sorry, for multiple years at all of our sites. And these are just two examples from two different sites that broadly span that aridity gradient. So we have Morgan Monroe State Forest, an energy limited system on the left here, and then we have an arid shrubland site on the right. And we see, for example, this coefficient value that's linked to the direct effect of GCC on bulk surface conductance is really high here, and it's much more diminished here in the arid site. By comparison, we see that precipitation is playing a much stronger role in influencing bulk surface conductance in these drier sites relative to its role in these energy limited systems. And so what we did is we essentially were able to use these coefficient values to estimate what's called the total effect of either precipitation or greenness on bulk surface conductance, and then look at that across the entire aridity gradient that exists at a continental scale. So the aridity index for each site is listed on the x-axis here, and then this left plot, we have the total effect of precipitation on bulk surface conductance, and then on the right, we have canopy greenness. And essentially what I just want you to show is that as we move from these wetter sites to these drier sites, we see a strong indication that the importance of precipitation is increasing, while we see the inverse for canopy greenness. And again, similar to what I showed with the previous study, the main point here is I wanted to provide this kind of very explicit example of how we're able to use the phenocam greenness indices in this case to better understand the role of phenology as a control of land atmosphere fluxes. And in this case, highlighting that given the coverage of phenocam, we can do it at pretty broad spatial scales and across broad spatial regions. So with that, I'll wrap up and I'd be happy to answer any questions. So phenocam datasets can be effectively used to address a wide range of scientific questions as it relates to phenology. The spatial temporal scale aligns with Flux Tower data products. I think I gave some good examples of that using both transition dates and greenness from phenocam. And then given the wide range of phenocam coverage and its co-location with the AmeriFlux sites, there are a lot of opportunities for investigating the role of phenology at broad spatial scales or across broad regions. And with that, yeah, be happy to answer any questions. Thank you.
Thank you, Adam. We have time for 2 questions. I have one question for you, Adam. Are there any important lag factors between the greenness index and the actual physiological changes or processes that are happening?
In some cases, yes. I guess maybe the first case that comes to my mind off the top of my head was that when we were looking in these arid regions in the Southwest, there was definitely a lag in most cases between when we see precipitation occurring, when we see evapotranspiration starting to occur, and then when we see greenness occurring. And we were able to discern essentially that during the monsoon season in the Southwest, you'll start to have evaporation before you have green up. There's a lag there. And you do ultimately have greenness, but it is less aligned and less correlated with greenness. And that's how we were able to determine from the second study I showed that it's a less important driver. It's more of a response to precipitation than driving changes in evapotranspiration.
Thank you. One more question?
So there's one plot showing the time series of surface roughness, and there are large fluctuations of that. So what's that? What are those fluctuations?
Sorry, which plot were you referring to?
Showing the seasonal roughness.
Oh, okay. This one?
I mean, I think this one, yeah.
Yep. And so kind of what's driving this? Is that the question?
Well, I mean, they're really fluctuated. So I'm just curious why.
Why our ultimate interpretation was that during, especially those shoulder seasons, where we were over predicting sensible heat flux, it was due to the fact that the canopy hadn't filled in yet at these sites. And so the roughness length for heat was effectively almost at the soil surface, especially during midday when the sun is kind of shining right overhead, while the roughness length for momentum is going to be much higher up in the canopy. And then so you're going to have a large difference between those two values. And then as the canopy begins to fill in, that roughness length for heat exchange is going to move. Essentially, it's going to be much closer to where the canopy is and where you have all your leaves and vegetation. And that's going to lead to this decrease or fluctuation in the difference between these roughness lengths or this KB inverse term. That was our overall interpretation, kind of looking at all the data and results.
Thank you.
Yeah.
Okay, our final speaker this morning is going to be Yuji Liu from Northern Arizona University, and she's going to be telling us about filling extra large gaps in eddy covariance data.
Sorry. I hope now it works. Thank you for having me as the last speaker, and I will be presenting our recent work about gap filling extra long gaps in eddy current measurements using a machine learning model. I will start with a brief introduction of our study site and then explaining the motivation behind, and then I will talk about the method to compare a traditional gap filling method with this machine learning method. And then I will show my results and wrap up the talk with 2 examples to apply the well gap fill data. So the study set is run, as you heard from Andrew before, it's Bartlett Experiment Forest. It's located in New Hampshire within the Sickle Ridge district of White Mountain National Forest. It is a deciduous broadleaf forest with mean stand age of 100 years and to 125 years. The dominant species there are red maple, American beech, and sugar maple. The climate there is humid continental. Actually, there are 2 eddy current towers there. The AmeriFlux one was established in 2004 until present, and the Neon one is started in 2016. The 2 towers are located 90 meters to each other, and the Neon one is about 10 meters higher than the AmeriFlux one. I think I have already talked to some of you that my current project is to compare the eddy current measurements from the 2 towers, but somehow before we really do a comparison, we came across the issue with extra long gaps in the data set. On average, after U-star filtering, we are looking at 66% of missing values in the data. It's worth noticing that after 2017, there's an increase in the gaps in the data, with some of the years going beyond 70%. And it somehow coincides with Andrew's move to Flagstaff. And unfortunately, after we are applying the marginal distribution sampling, the standard methods, the long gaps are still there. There are 2 extra long gaps marked in blue here. There are about 170 days and 200 days. That's crazy long gaps. So now I would like to recap a little bit more about the marginal distribution sampling. It's a standard gap filling method uses FluxNet and equals. It works by seeking the meteorological conditions similar to the ones for the missing values. However, the performance of the MDS for extra long gaps is limited, and there's also a paper published earlier this year reveals that MDS causes systematic bias in carbon balance estimates. And for CO2 flux, there are typically 3 predictors are used. They are short wave incoming global radiation, air temperature, and VPD. However, using more than 3 or 4 drivers is not generally recommended for MDS. Considering all these limitations, we are trying to explore a method, and the tree-based method has been reviewed with good performance for in gap filling CO2 and methane data in previous studies. Another good thing about machine learning model is that it's possible to include more predictors. So in the end, we use GCC, EVI, air and soil temperature, PPFD, soil water content, and VPT all as the predictors in the model. And for the workflow, we started by removing some outliers from the raw data, and then we estimated the contours of the U-star threshold distribution. For each U-star scenarios, we filled the gaps using MDS and XGBoost respectively. In the end, we compared the results. I think Andrew and Adam, they already talked what is GCC and how they applied GCC in estimating ET I want to add here, there's a previous study show that canopy greenness is highly correlated with the rates of photosynthesis in deciduous broadleaf forests, but it was not used in gap filling yet. So before hyperparameter training, I did a pre-test using a new Python package. It's called Lazy Predict. Why is lazy? It's because probably you came across the question, what algorithms to use in the big machine learning world. And this package provides a solution with which is really fast. You can select your models without hyperparameter tuning, and the result shows attribute descending here. So we finally decide to go with it because of the speed, and it's almost as good performance. Here are the results. The NE gap filled by attributes in orange successfully cover the gaps which cannot be filled by MDIs. Regarding the model performance, compared to MDIs using attributes result in improved model performance as indicated by lower RMIC and MAE, as well as higher R-squared. While MDIs shows very small negative bias, yeah, here. Here's the result about annual sum, and the arrow bar here is the standard error. Methods. The difference occurred for the summertime. Using attributes consistently indicates a smaller carbon sink compared to using MDIs across years. Here I'm showing the results for the future importance. Actually, the heat map is about 4 models, 1 model for each column. I'm doing so because I want to know the role of GCC if it's really improve the model or how important it is. So basically the four models, they are like including or excluding GCC or EVI in the model. So, but there are some consistent results. I think I lost my connection again, but I'm okay too. I think what you can find here is PPFD diffusion. PPFD acts as the most important predictor here. It can be explained because diffuse PPFD allows for the even distribution of light for the canopy, so therefore enhance the overall photosynthetic efficiency. And the GCC here, it works as the second most important predictors. So that's what we expected. As we explained before, GCC reveals information of canopy and vegetation changes that is not included in other meteorological variables. Okay, they're here. And for the last column, that's a model that without GCC at all, we have EVI instead. So in this case, EVI also has a high feature importance. Before the end, I will show you two examples why we really need the gap field data. Here is a part of my future work to compare the data for pair towers, the AmeriFlux one and the Neon one. So the XGBoost results are showing in darker colors. It's possible to make a comparison now because with the outliers in annual sums from MDS, there are some extreme values, so it was impossible to make this comparison. Actually, we are expecting better agreement for the or the landscape, but somehow we are still seeing the differences in the measured fluxes by two towers. And in EDU, I'm trying to, I will dig more about this issue, and I will try to present more about evaluating the continuity of the two data stream at EDU. And another example is from my, it's connecting again. I'm so sorry for that. I think I lost my connection to the Zoom meeting. Yeah, it's possible, but it's really a pity if I'm not showing the figure. But okay, I think I can do it. Because another work is from my colleague Darby Bogle, and she wanted to synthesis the long-term data for Bartlett because it was established in 2004. So we have a long-term record, which is a very valuable data set. So she used XGBoost to fill the gaps, and she found a clear trend of decreasing in long-term carbon sequestration for about 20 years in Bartlett research forest. And then she did something very interesting. She plotted, let's imagine, She plotted the monthly sum over years in one figure, and for the XGBoost results, she found that there's a clear trend towards smaller carbon sink for recent years, but the pattern were messed up for MDS results. And then my next slide you are not seeing, but I have it here. It's the most important takeaway message. Okay, so I would say the model performance of XGBoost is better than that for MDS, especially in the context of extra long gaps. And it's probably because more predictors can be used and there's no assumption on the light response curve. And XGBoost provides reliable estimates of the annual sums of NE. It is particularly useful in terms of the study like site comparison for my project and the long-term data synthesis for my colleagues. So, and yeah, that's close to the end of my talk. Just to thank all the co-authors. They are listing in the slides you are now showing. They are Jeremy, John, and Jingjing. This work is supported by National Science Foundation. Thank you for listening. Thank you for your attention.
Hi, first, thank you very much. And I like just hearing the take-home message. So that was great.
Yeah, thank you.
I know this wasn't part of your work, but do you have any thoughts or insight about whether XGBoost might be useful for shorter gaps and how using it might compare to the MDS or other methods? Thanks.
That's something actually we were expecting, like for shorter gaps, we expected similar performance or similar annual sum estimated from MDS and XGBoost. But the thing is, they still have like XGBoost tended to estimate a smaller carbon sink. So that means for shorter gaps, they don't have very good agreement. Yeah, that's my answer now.
So a quick question and comments are, so when you show the cumulative sum per month for those two approaches, boosted and the MDS, it's very different. I think the MDS provides also fairly large error bar for Bartlett specifically. So I think you should show this also because they look very different and scary a little bit for somebody that wants to look at cumulative sums. One thing that you might have done is, have you looked at long time series at other sites, Harvard Forest, UMBS, when you create artificial gaps and see how the method perform at those sites and see? I think that would be great.
Okay, so sorry, I don't fully get your question.
The first is a comment on you should really show all the error bars from the MDS method when you compare the annual, like monthly.
Yeah, exactly.
Not the one that you had, but
yeah,
okay.
I know that. Thank you.
And the other one you should, have you tried to use long time series at other sites, create artificial gaps and use your method, the two method, and see how they perform? Because I think Bartlett between Neon and Bartlett site is a little bit difficult because of the heights difference.
Measurement heights are different. Yeah.
Have you done this?
Yeah. I think the measurements heights are different, so that's probably the reason why I still see the disagreement in the measured fluxes for the annual sum. But in terms of the gap filling methods, I have the results for Neon, so I trained the model respectively for AmeriFlux and Neon. So that's what I am doing. Does this answer your question?
Thank you. Let's thank all of our speakers this morning.